Yeah, thanks. Thanks everyone for coming again. So this is the last chapter of my thesis, and actually, some parts of it are kind of under development. We're sort of expanding things a bit, and we're going to resubmit it. So yeah, I sort of wondered whether or not I'd have some of that done for you to present today, but it's not quite done. So expect an update on the archive version, which you can find through Discord most likely. This is joint work with Erdwansha in Indonesia. I think most of you here probably are familiar with geometry and topology, but yeah, so we may not need to go over this second slide here. But topology in general is about counting the number of holes in things. For example, a donut and a coffee mug have the same number of holes in terms of like where you can stick your finger through the middle of the hole in the donut or in the handle, right? And convexity will come up also in this particular work, where convexity is just if you pick 2 points within the sort of shape, for example, if you draw a straight line between those 2 points, the shape is convex if that line is completely contained within the shape, whereas it's not convex if that line is partly or completely outside of the shape. So that red part, for example, in the line and the non-convex shape is meaning that this whole shape becomes non-convex. And algebra, well, there's different ways to think about it or to explain it basically, but my favorite example is like a Rubik's Cube. So you have the set of all possible states of the sides of the Rubik's Cube, and when you perform an action, for example, you pick it up and you put it down, well, you didn't do anything, but for example, but if you count that as an action, and if you also count like making different twists and turns as actions, then starting at a particular state and following those particular rules gives you a particular set, and that set is like all possible things, and you have rules about how you can get to possible things, those actions. So this whole object, this Rubik's cube, and the things you're allowed to do, the rules of the game, sort of give you this set with particular structure. And we'll talk more about how you can get similar sets with some sort of structure in terms of the neural code. And the analogy I want you to think of when we get to that is a bit like playing with a Rubik's cube, where there are some states or moves or patterns which are sort of banned. So I'll get back to that analogy a bit later. But first, let's talk a little bit about neuroscience. And in particular, let's talk a little bit about the hippocampus. So the hippocampus, if you don't know already, is a place in the brain, sort of in the middle of your brain, which is heavily involved in both memory and navigation. So in terms of navigation, for example, we've detected these things called head direction cells. So these head direction cells, for example, were first discovered by basically strapping a rodent in place with their head faced in different directions. Pardon me. And you can see those directions marked in this box on the left, and those different arrows are pointing with the letters is the different orientations of the animal. And then what they noticed is that for one particular cell in the hippocampus, it was active more when it was so this axis here is time, and this is sort of time spent in A, and also here in A, and then this is in B, and also in a little bit in C. So they were sort of moving around the animal, and they saw that it was quite active when the animal was orientated towards A, a little bit towards B and H, but not towards anything else. So this was a head direction cell. It's selective for a particular orientation of the animal. And you also see things like place cells. So this example here in the bottom left, that's a cell that's we're looking sort of top down on a circular environment, and this heat map is showing where a particular neuron is active in place. And these ones sort of on the sort of middle that I'm just putting a box around, these are called grid cells, and these are shown to be in an area adjacent to hippocampus, which have this grid pattern. And there's lots of different theories about how they interact with place cells and head direction cells and other types of cells, and which ones are sort of the the basis, or which one is sort of the most fundamental unit, or how do they interact. There are lots of theories about this. And there are some references here on the left if you're interested. And ultimately, it all comes from a hypothesis put forward by Tolman, which is about cognitive maps. The idea being that animals and indeed humans create these cognitive maps in their brain to navigate in the world and in social situations and in all sorts of things. And more recently, that is in the past, say, 50 to 100 years, well, even less than that, people have been thinking about and knowing more about the role of the hippocampus in memory as well. And one of my favorite researchers on this topic was Howard Eichenbaum. Unfortunately, he passed away a few years ago, but he was sort of famous for doing slightly unconventional experiments, at least according to the status quo of hippocampus experiments. And for example, he found a whole bunch of sort of things that that cells in the hippocampus coded for, which weren't really anything to do with navigation. They weren't the traditional sort of place cells or grid cells or those sorts of things that people were famously finding. He was finding things like time cells, like the things that sort of like will be active for when you're in a place for a particular amount of time. It's sort of like counting or something, but also these sorts of relational things. So take this example here of the rodent. It's given two different sort of containers which have a particular sense, and there's a relationship between these containers where it's shown only pairs which are adjacent in the sequence A, B, C, D, E. So it's shown AB, and then BC, and then CD, and then DE. And it's taught that the later of these in the alphabetical sequence has a reward sort of buried just underneath the foot of this little container. So it would sort of dig and find the reward. And it's given lots of presentations of this so that it learns these associations, such that, oh, and sorry, maybe I did it the opposite way. I think it's the earlier letter. So A is greater than B and B is greater than C and so on. But it's never seen, for example, B and D together. And so there's a question about whether or not it can make the inference that, based on this sequence, that these sequences or these relationships it's already seen, can it make the reasoning, can it figure out the fact that actually B should be greater than D, even though it never saw that pair? And in this classic experiment, what they did was they had three different conditions here shown in this little these three little sort of shaded keys, and in the second two, well, this one, the full one, is a healthy animal, and in the second two, they basically destroyed a little bit of the connections and the tissue in the hippocampus. And what they showed was that if you destroy just those specific places, you destroy this longer sort of relational inference. So the BD performance goes down to chance, whereas the initial things that you sort of were taught, those initial relationships stay high in the healthy animals. And then those initial, these things here, which are these, still perform well. So you're still able to sort of memorize things, but you can't make these longer term inferences over the structures that you memorized. And so these areas of hippocampus were then studied more to figure out what are particular cells in this area that were related to potentially these inferences. And subsequently, there was a hypothesis that started forming about, well, maybe all the hippocampus does is not necessarily, and there are specializations within hippocampus, and there are different areas and subregions, but maybe the ultimate function is not necessarily to do with space or memory or navigation, but rather something to do with sequence prediction. And so I really like this quote, and I think Eichenbaum was quite ahead of his time. This is from 1999, and he said that, or with colleagues as well, that coding of spatial locations by hippocampus place cells emerges from a fundamental representation of behavioral episodes. And then in this little picture, you can see sort of a cartoon of a behavioral episode where the rodent starts here in the center and moves towards one of the, in the case of his experiments, it would be like BD, for example. It can't thing, so no, that's not the right one, comes back and then goes here. Or you could think of these as different episodes, or what have you. But he sort of said, well, these, in order to learn these sequences, or to learn these behaviorally relevant or ecologically relevant outcomes, for example, to get the reward, you just naturally learn sequences of actions, and those actions, for example, cause a representation of place. And sorry, just a moment. Sorry, actually, there's some helpful gentlemen here to clean my windows. Okay, sorry. Yeah, let's keep going, maybe. So. Okay. All right. So I sort of mentioned that Eichenbaum and Tolman characters, and that they were sort of maybe doing something which is ultimately similar. And some other researchers have recently sort of moved on to sort of formalize this, and there's something called the Tolman-Eichenbaum machine, which maybe some of you have heard of. I think Dan definitely has. And this is by a group at Oxford, and they notice that the following things are sort of similar. So take, for example, all of these different graphs on the top row, so sort of panels A, B, and C. You'll notice that if you have some understanding about what these graphs are, just intuitively, then you'll know that, or you'll notice that if you know what the blue edges are, you can infer what the green edges are. So let's just take A as an example. So if you know that Jane has a relationship with Alice such that Alice is Jane's mother, and you further know that Bob is Alice's brother, then from the perspective of Jane, then you know that Bob is your uncle, for example. And the same thing goes for space. So if you go east, south, west, and then you know that north must be here, for example. Sorry, was there a question?
No.
No, I just thought it was funny that you said Bob's your uncle. Good choice of
Yeah,
words.
I said that on purpose. Yeah. I think they did it on purpose as an Easter egg. And so what they did was they had this task where they had to predict the next sensory object, and they had different sorts of sort of structures of the environment. One was this grid structure in D, another was this kind of family tree structure in E, and then I think they also had, like, if I recall correctly, like one that was made up by triangles, kind of like this. I've forgotten now. But the task is, given this model that they made, and given a sequence of actions going, from one vertex or node within this environment to another, and then given the sensory outcome, you're asked to predict the next sensory outcome based on a given action. And so here we can see, well, if we moved right and we saw the bicycle, then we must have probably come from the banana, if we have knowledge of the world here, as we do. But the model doesn't see that. It only sees this in the red box here. But if we followed this through, well, we go right to see the book, we go down to see the table, we go left to see the chair, we go up, we should see the motorbike again. So the motorbike should go there. And then the same sort of thing goes through for these family trees. And what they showed was that based on these sorts of relationships, you get the appearance of place cell. So you train a model to do this task, and then you look inside that model to see where its activities are showing up in different environments and so on. And what you end up seeing is things like grid cells and place cells and even time cells and all sorts of things. So you get a sort of a nice confluence of different ideas from both the navigation and memory sides from people like Tolman and people like Eichenbaum in this machine. So that's quite nice, and I recommend reading that paper. It's quite influential to me as well. Now, people might...
Actually, sorry if I can ask a question. So
yeah, go.
I read that paper, I think when it came out or even beforehand, and I liked it, but I don't have enough of a window into the neuroscience community to judge how it was received. Is there a consensus that this is... I never quite know how seriously to take. There was a few papers of which this is kind of one, which were showing the emergence of grid cells or place cells in things that look like reasonable models of hippocampus. And maybe this was... the Tolman-Eichenbaum machine in that paper is relatively complex compared to some of the other models that were employed in those other works. And I wonder what the takeaway for the community was from... what do people infer from this? People sort of buy the story, or is it still maybe unclear?
Yeah, that's a good question. And I would hesitate to speak for an entire community, of course, but I think there's a mixture of opinions. So some actually think that it's just an extremely fancy, hidden Markov model. There's also others that say, no, there's enough structure here or constraints put into the model that it's a meaningful sort of representation of our mappings between what we know in neuroscience, like anatomically or functionally, and what we can see behaviorally and these sorts of things. They did also, I think, include an experimental sort of they collaborated with some experimenters in New York, I believe, who also did some nice work to sort of verify their model. I would say in general it was quite successful because it sort of had some famous people attached to it, and it was like a nice result, and it got in a good journal, and it was a really good result, and it is a really good model. But also it's not the first model to do things like this, nor is it necessarily the first to sort of combine these things, but I think it did a great job in popularizing and combining lots of different ideas into some single thing. I don't necessarily agree with all the choices or commentary about it, but I think it's an extremely good model and very interesting.
Thanks.
But yeah, I hesitate to be too critical about it because I think it's actually quite good. But still, it's Anyway, there's a lot of things I could say.
I guess the critiques maybe seem to me like critiques you can level against almost any systems neuroscience kind of work that's putting together models of different pieces.
Yeah, exactly. Yeah. And there is, there's always going to be also a certain crowd who are just, anti-theoretical or computational neuroscience, unfortunately. Yeah. Hope that's enough of an answer, at least for now. Okay. So, all right. So I just wanted to mention as well, like you might have a question about, well, are they like, what do these neurons that are like place cells or so a neuron is a type of cell, a specialized cell in the brain, and the main one that people study, even though there's plenty of other types of cells, but functionally they're very important, and you might want to know, what do these look like, or what do they do, or why should we care? And also, are they sort of individual? Like if I knock out one of these place cells, is that just like place not represented anymore? Well, it seems like that's not probably the case. It seems like there are so-called assemblies of excitatory neurons. Excitatory just means the neuron is sort of sending a positive signal and sort of saying, hey, look at me, I found something out. How about you take a look? I suppose negative signals also do the same thing, but just the inverse. But excitatory assemblies or groups of neurons are quite well known, especially in hippocampus. So CA1, this thing at the top left of the slide on the right, CA1 is an area of hippocampus, probably the most well studied. And here you've got 5 different electrodes picking up electrical signals from these neurons, which are separated by, I think, 50 to 100 microns each, which is quite a long distance in the brain. So then what you have is this raster plot showing times on the x-axis. So this is time. And then on the y-axis are these different units, functional units, which we think are excitatory neurons in this case. And what we see is that they cluster together in these so-called assemblies. And those assemblies are functionally picking up on something similar in the environment, potentially. So, for example, they might be picking up a lion or a leopard or a tiger or something. And to the extent that they have similarities with one another in terms of the identities of these neurons here, for example, they might share some coding, some features of both the tiger and the lion, whereas these ones on the right side, those particular neurons might share some information or some coding of both the leopard and the lion. And so there are these kind of, this interesting mix between continuous and discrete, approximately continuous, but very much discrete groups or assemblies of neurons which sort of collaboratively encode information, and they're spread out over distances. So we have some guarantee that if you just knock out one of these neurons, which can happen through lots of reasons, then you're not going to lose an entire place or memory or something like that. And so these relationships and these functional maps, these cognitive maps that you build, won't be destroyed too easily, but they can drift and change with experience. So one question is, and the question that we have for this work here is, how can we infer or build some kind of map? For example, like on the left, these sort of discrete maps with like family trees or environments. How could we discover such a map by only looking at the activity of neurons? So let's move on to the next slides to discuss that. So on the left, you'll see, on the left of the left slide, you'll see 2 sorts of columns. I'm just going to draw those 2 columns here. Okay, so let's call example number 1 and then number 2. So in example 1 and 2, on the top row, you've got these circles. Those circles are receptive fields in some abstract space, and here, we can think of, for example, a 2D environment, for example. So you might think that these are tiling or slightly covering, not completely, some square environment, for example, and we're looking from the top down. And in this case, there's like a hole in the middle of this environment, for example. And then these different numbers represent different neurons that might be part of different assemblies of neurons. But we can just also think of them as individual neurons. And then you'll see that these sort of looms and these different intersections between the convexly shaped receptive fields represent these intersections of those. And then what we do is we basically just look over time. So here's on the second row, this is our time axis on the x-axis, and then our different neurons on the y-axis. So it's a little bit like that raster plot you saw just before. And these different vertical lines indicate when that particular neuron was active. And then what we do is we organize these kind of vertical slices across a particular time range or a time bin, and we just look at which neurons were active in that time bin. And if they're both active, then we make the inference that, or the guess, that those two neurons have an overlapping receptive field. Now, the reality is that it's a bit more complicated and that you have to make certain assumptions in order for that to be true. But this is the basis of basically everything that follows. We basically are looking at coactivation of neurons, and then from there, guessing the structure. So the structure that we can build from these two examples are these little simplicial complexes. And what you do is you say that, well, if you see two neurons together happening at the same time, then that corresponds to a one simplex that is like an edge in a graph. And if I see, if in a simplicial complex, you have the condition of downward closure, and so you know you must have both 1 and 2 as 0 simplices or vertices. But then you also do the same thing for all these other slices of the time bins. And for example, this one here corresponds to this triangle, since we have 2, 4, and 5 all active at the same time. So we have this triangle here. And you'll notice that I've written beta 0 and beta 1. These are the Betty numbers. So if you do simplicial homology, you can have a formal way of counting the number of holes in this environment. So it's about the topology. Betty 0 is the number of connected components. So here there's only one connected component. If there was, say, another neuron out here, number 6, and that just fired on its own, for example, if we had a 6th row here and there was a little bit of activity here, but it was just empty for the rest, and there were no other neurons in this final time bin, then we would have Betty number 2 in this case. But we don't have that, so this is just one connected component. And then this Betty number 1, beta 1, is equal to 1 because we have this hole here. And if you're interested in how to calculate that, then you can look up simplicial homology. And then the same thing happens here on the right.
Can I ask a question about, I presume that when you find these fields in order to make measurements like this in practice, there's a little bit of circularity here, perhaps, where you use your knowledge of the topology of the environment in order to figure out what a plausible candidate for the relevant receptive fields are? Or am I wrong about that? Like, how do you determine, if you're actually to do this in practice, how would you somehow know? I mean, if you want to verify that this is actually a reasonable way of interpreting what's going on, how do you determine where to measure, which neurons to measure?
Yeah, that's a good question. So you have to, so in the case of, so I should say, yeah, first of all, basically most of this work and this sort of like building from this basic sort of setup has been done on toy problems where the data is generated by some, theoretical means, and it's sort of constructed to give you a nice answer, or a meaningful, mathematically or some theoretically meaningful answer. There has also been some empirical work, but I say empirical, it's not actually from data as far as I'm aware, but rather from sort of simulations of data. And that's what we also do later on, which I'll talk about. But fundamentally, what we've done is we've sort of introduced this new algorithm, which makes it so you can now do this on sort of real data, real quantities of data, because the previous techniques were quite inefficient. And so there hasn't yet been rigorous testing empirically about how to go about choosing all these neurons and so on. One sort of initial thing that I'm interested in is, well, obviously, neuroscientists and others have lots of ideas about, well, for example, hippocampus has this particular function, or, represents this particular set of stuff. And so a natural thing to ask is, well, okay, let's look at all the neurons in the hippocampus, and then let's look at this, make these sorts of simplicial complexes or relevant structures and count the holes, or, do stuff like that to see if it actually maps to what they say it should map to.
Mm-hmm.
And the same thing is we're thinking of doing in the visual cortex as well.
Cool, thanks.
Yeah, no worries. So, as some of you might know, if you've worked with simplicial complexes and complexes, and you've also worked maybe with some computers, you'll notice that to store and to do calculations with very high dimensional and large simplicial complexes is quite difficult. You'll also know that putting it into different structures or things like that, different other forms of structures, also doing some kind of compression can help, but is not perhaps the whole answer. But the first step to doing this, and then also to getting some sort of back doors to different questions about, like, for example, the number of holes that might be in this structure, is to actually encode these simplicial complexes algebraically. So I'm actually just going to tell you some examples and what they mean sort of intuitively, and then we're going to sort of go back, sort of like go backwards and reintroduce this more formally. So there are two main important sort of relationships that we might care about in this case, and those will be called monomials and mixed monomials, which are these things here. So this is the monomial and this is the mixed monomial. And these are relationships between different neurons, and they live in this structure called J sub C. J sub C is called the neural ideal, and we're going to formally introduce that in a moment. And if you see these different monomials living in J sub C, then it will imply something about the receptive field relationships and vice versa. So U are receptive fields, and sigma is some set. So for example, if you see the monomial X subscript with this sigma set, it means that all of those different receptive fields in sigma are disjoint. So their collective intersection is empty. And this is sort of, this here is sort of like just a notation for this.
I can't hear you, Tom. I think your audio cut out, or maybe mine.
Yeah. Sorry.
You're back. Yeah.
Is this better?
Yep.
Okay. Yeah. So did you catch this thing about notation?
The beginning of it, yeah.
Is just my notation for example, in this case, xi, xj, and that just means some relationships between these two things don't exist. And then this one here is the same thing, this little guy, x sub sigma. And a mixed monomial, as opposed to a monomial, is about subset relations. Sorry?
I think I'm a little confused. So sigma stands for a set of neural fields, right? So in this case, a set of integers between 1 and 5 with 2 or more elements, or is it always 2?
With at least 2. Yeah, 2 or more. Yeah.
Okay, so I take a subset sigma of the set of integers 1 through 5, or however many neural fields there are, and if there are no overlaps, then I form this product of variables standing for fields, and I add that relation to my ideal. Yeah.
Yes. Yeah.
Okay.
And that relationship is a monomial relationship. And then we have these other ones called mixed monomial relationships. And that's where you have two such sets, sigma and tau, which are different. And this, these are also, so sigma could be a singleton now, and tau also could be a singleton. But so this is kind of like one or more. And now you get relationships like, for example, let's call this one I and this one J. Now you get relationships like maybe I don't use I and J because it's kind of, I'm using I's in here. How about let's use K. So then, yeah, that's right. So this relationship means that that K, this example I've just put as K, is whenever that's active, whenever sigma, or in this case J, is active, K is necessarily active as well. And you'll sort of get, but for those of you who might already know what are ideals and things like that, basically these are zero elements, and we have a finite field of F2 in this case. So you might, if you're thinking ahead, that's why these have this structure, like 1 minus and so on.
So in particular, I have xj times 1 minus xj. Is that right?
Yeah, that's true. But that's trivial, right?
Yeah. Right.
And so this ideal, this neural ideal, tries to, well, it gets rid of those trivial ones, and then we have something called the canonical form of the neural ideal, which is sort of like you just try to make it minimal in the number of elements.
It's a reduced Grosvenor basis.
From this minimal set. Sorry?
Meaning a reduced Grosvenor basis, or there's some other notion of minimal?
Yeah. Well, yeah. Maybe it's a reduced Grosvenor basis. Maybe you can tell me, actually.
We'll see. Maybe.
Yep. All right. So this is just some sort of like intuitive introduction to what you're going to find out about this neural ideal. But for the people that maybe haven't got a working knowledge of these things, or just need a refresher, I'm going to sort of try to quickly go through some of the building blocks to get to this neural ideal. So the first thing I need to do is tell you about polynomial rings. So polynomial rings are a special type of a group, right? And what is a group? Well, it's a set of objects, a bit like, we talked about the Rubik's Cube at the beginning, and that has particular properties. So all the elements in that group, along with different, I talked about different actions, but you could think about basically different operations. For example, in this case, plus, or some multiplication or something like that. But also you think about divisibility or other operations. And the idea is that you have some set of these things along with the operations that satisfy different conditions. So closure is the idea that once you perform all the different operations, you have this sort of fixed set of things. Well, yeah, I suppose it could be an infinite group too. Associativity is about the rules that do your operations work together in certain ways or not. Identity, well, you have to have some sort of operation or an element mixed with an operation where you, given that operation and that identity element, that you get back the same element. And invertibility is the idea of, for example, multiplication and division. And then you have other special types of groups called abelian groups, where you have an extra condition. So this is still a group, but now you add some extra condition onto it. which is commutativity. That means that you can move things around, like you can change the order. It doesn't matter so much anymore, or it just doesn't. And then you have other things like free abelian groups, where you care about particular bases. And then you also have rings and fields. And I've sort of tried to make this diagram to be that, to be sort of just visually descriptive, roughly, of what, so all these things on this picture are a group, but only those contained in this green circle are abelian groups. There are some rings which are non-commutative, so that's why there are some outside of this green circle. There are also free abelian groups and fields and lots of things like this. A polynomial ring is going to be usually commutative, right? So it's normally going to be living in this green circle, but it can also be out here. It has two operations, and K, this thing, so this is how you denote the ring. K is a field of coefficients, and X is a set of variables. So the elements of this are called polynomials, and they have a form, generally speaking, like this. So P0, for example, and then there's sort of a hidden X here. You can think of it, but it's just 0. And then that you can also think of like this as X1. And this goes all the way up to M for some order. And these P's live in K, so those are the coefficients, right? And the X's have this regular exponentiation. Now that's just theoretical. Well, not just, but that might be too theoretical for some. So let's look at an example with apples. And actually, I was quite pleased when I was walking around before. I actually picked up an apple just near here, and I wasn't able to keep it because I clicked on it, and then my character ate it. So I was kind of disappointed that I didn't get to keep my apple. But anyway, so here, but I picked it up right here. So okay. So here's an example.
Chad's come back with some apples in his hand.
Oh, good. Yeah, good. Good. So let's say that apples, like just one apple, is our X, right? And so remember, like the polynomial ring is like this, right? So what's K here? Well, K is R. And then our X is an apple, a green apple in this case. So here are some elements that are in R apple. I don't know actually how you say it. How would you say that? R on apple? R over apple?
The free polynomial, the free ring on apples.
Yeah. So here's some different elements. So Q1, Q2, Q3, Q4, these are different elements. They haven't got any particular structure to them, they're just examples, right? But then what I'm doing is showing here some addition operations of those. So let's just do the first one, this one of Q1 plus Q2. So Q1 has a 6 and Q2 has 7.2. Okay, so we just add those numbers together. We have 13.2. And okay, yeah, Q2 doesn't have anything else, right? It doesn't have any of the special sort of apple things. So we just add those together directly from here. All right, let's do this second case, Q3 and Q4. Okay, so we have a 1. But you notice we don't have like something to like the 0 apples or something like that, right? So then we just put the 1 here. And then we need to match up basically these.
What's up with Q3 times Q4?
Did I get something wrong?
I don't know how you get 6.4 apples squared.
Let's see. Yeah, I don't know either. What was I doing then? Maybe I was doing a different set of variables.
You might have changed the examples or something. Yeah.
Yeah, but yeah, I mean, let's just do that for completeness though and correct this. So Q3 times Q4, so should be, well, these are the only two things that are in common, right? Actually, yeah, maybe that's what I was thinking.
I don't know what operation you're doing. This is just multiplication.
Yeah, just multiplication, right? So then, isn't there a 0 here?
Oh, I see. Okay. Well, that's not how you multiply polynomials.
Oh, okay.
So you're multiplying them basically like taking a dot product, right?
True. Yeah.
Yeah. So you want to basically view the Q3 as a sum of two terms, Q4 as a sum of three terms, and expand the brackets. So you'll get.
I see.
Yeah. So neither of those examples is probably correct, I think.
Yeah, okay. So yeah, please ignore this then. We can maybe come back and correct these later. All right. So yeah, that's yeah, let's just pretend that those examples were correct and trust me for the rest. So the neural ideal is defined on the right slide. So we have a finite field F2. F2 just means, for example, 0, 1, like this. And n, well, n is our number of neurons. So we have the finite field over all these variables here. So this is our polynomial ring. And then some characteristic polynomial on V is defined like this. So what is V, by the way? V is like, for example, where n is equal to 4. This is an example V. This should be, yeah, like a vector. And then we plug it into here. So the support of V is going to be equal to the indices. So 1, 2, 3, 4 is going to be equal to a set. Oh gosh, a set of the indices that have a 1. So you can see 2 and 3 both have a 1. So therefore the support of this vector, this example vector, is 2, 3. And so then we have these two different things. So for I in the support of V, we'll have, so our characteristic polynomial for this V is going to be x2, x3. And then for the j's, which are not in this, so that means 1 and 4, 1 minus x1, 1 minus x4. It's going to be like this. And C, small c, is a code word. And you can just think of that as basically just another name for this vector. That vector is just a code word. A code word being like when we had that table thing with all these different columns where some of these were active and some were not active. That row is one of these code words. And then this whole thing is the code. So this whole table or the collection of all the rows there, all the code words, all those vectors is the code. And this thing here is, yeah, that's the code. So it's a subset of this. And then you can create something called the vanishing ideal. The vanishing ideal is defined like this. So f is some polynomial, some polynomial where it will be 0 for all the code. And so they vanish, so it sort of vanishes on the entire code. And then the neural ideal is just the characteristic polynomials for this. So you can say that J sub c is generated by the characteristic polynomials of the non-code words. That means that if you give any code word vector to this J sub c, it will be 0, and it will be 1 if since these are all binary variables, all of these things will give you like a 1 or a 0 if you give it a vector, right? And then it'll be 0 if you give it something from the code, and 1 if it's not from the code. And this is, roughly speaking, where I get that analogy about like there being some kind of like band moves in the Rubik's Cube. So it's really just kind of identifying things which are in the set or not in the set, and sort of like bad, well, not necessarily bad, but yeah, things which we're not, our system doesn't show in the case of the code words. And so those code words, if you are bringing those from a simplicial, well, you can also build a simplicial complex, and as we'll see, there's like a connection that you can make between this structure and the simplicial complex through a different kind of ideal that you make from. This is basically a modified version of something called the Stanley-Reisner ideal, which encodes algebraically in a similar kind of way the simplicial complex. Did I explain this correctly or well?
Sounds right to me.
Okay. So I'll go on to the next. So once you have a neural ideal, you'll notice that you have way too many polynomials, right? There'll be basically a lot of redundant polynomials that just tell you the same thing as some other polynomial does. And before we sort of notice which are the essential ones, which could give us all the information that we want to know about like monomials or mixed monomials and things like that, like I talked about before, we can first sort of identify something called pseudo-monomials. So this has a general form of this. And this is where those sigma and tau sets come in, and the intersection between them is empty. And these two, number 1 and number 2, the monomials and the mixed monomials you saw before. The negative monomial is, or pseudo, is something like that will only appear when there's a code word which has ones the whole way through. So that one we don't really, well, it exists, but it's not. It's sort of topologically speaking, if you go back to this simplicial complex, it implies that we just have, pardon me, a point. So it's kind of sad if that appears. But notice that because of this condition here, this intersection condition being empty between sigma and tau, we don't get this example that Dan mentioned earlier about like, what if I have xj times 1 minus xj or something like that. That's not a pseudo-monomial. And so we construct the canonical form of this thing, J sub c. So we first make J sub c, which is like all of the pseudo monomials, F monomials. And what we want to do is we want to make sure that there is no other pseudomonomial such that the degree of G, this other pseudomonomial, is less than the degree of F, such that if you compose those, there exists some other polynomial H. So if you compose G with H, so it's kind of like factoring. And then what you're left with is called the canonical form of J sub c, and you denote that with capital C, capital F of J sub c. And this is the set of all minimal pseudomonomials in J sub c. And for example, if you have J sub c as x1, x2, x3 as one polynomial, and then x1 times 1 minus x2 times x3, you can actually just have the shorter version, which is just x1 times x3. That will give you the same elements, right? So let's do maybe an example in the form of like asking, well, what does this give us even once we've constructed these things? So one example application is convex realizability. So if I have, this is my code here, so these are vectors, and for example, this, the support of this is like this is 1, right? The support of this one is empty, and so on. And then we go through that process of creating the pseudomonomials, creating the neural ideal, and then looking at the canonical form of that. Well, the elements, just by looking at these elements, because of the way this is constructed, we can infer something about the relationships of the receptive fields to one another. And you'll notice in this case that it can't be true that these receptive fields are convex because of the following two facts. So firstly, this polynomial exists. And what does that polynomial mean in terms of receptive fields? Remembering that U is like a receptive field. That's like an area where a neuron becomes active. So what this means is that, well, if x1 and x2, those variables are active, so those are the neurons for x1 and 2, then x3 and x4 can't be active at the same time, right? So that means that so the intersection of 1 or 2, sorry, I said it the opposite way, has to happen at the same time as x3 or x4, right? So that means that the intersection of these two, if we draw a little picture, this is just one realization. We could have others. Let's say this is 3 and 4. And then, oh no, I wanted to make that bigger. So let's make it, like this, 3 and 4. 1 and 2 are inside of these, so 1 and 2 are inside here, let's say. But because of this, x3, x4, it means that x3 and 4 doesn't have, like its intersection between these two receptive fields is empty. So that's not possible with this picture if these are convex, but it is possible if you make it non-convex. So and then this is just written. And this situation is called a local obstruction to convexity. So maybe it's a
really
good time
understand
to
the
just pause here.
emphasis on convexity. Like, why do I care about whether the neural field is convex or not?
Because they typically are.
I see.
Not always, but they typically are. And I think there's something like 10 or 20% of them are non-convex, or approximately non-convex. We don't have great statistics on this, but most of them are, and most of them, if they're not empirically, then functionally they are. Like, and empirically meaning kind of like if you just look at the receptive field shapes, they might be a little bit wonky and non-convex in places, but they function as if they are convex, it seems, or at least that's a hypothesis. And it could be the case that that you have a set of neurons, empirically speaking, which do show non-convex receptive fields, and in that case, then that's interesting, maybe. So yeah, that's basically why.
But I guess there seems to be some reason to think that the convexity or lack of convexity is meaningful in some way. It's not just like some accident of the arrangement.
Yeah, that's right. I mean, and also too, if you can ensure that the code is a convex code, then it's easier to do some checks for other things. Like if you make the assumption that your receptor fields are convex, then you can have better lower bounds for like the number of dimensions that these things must span and things like that.
I see. Just to let you know that we're sort of over time already, and we have a hard stop at 4, so maybe we want to get through. Let's make sure we don't miss out on the good bits.
Yeah. All right, let's go ahead and skip a little bit. Yeah, sorry, I have a habit of going over time here. All right, so let's just go to this very last slide here. So just know that there are other applications, and also that there's this thing called the Stanley-Reisner ideal, which is basically where this kind of comes from. So the applications that you can have basically to get topological and geometric information, and there are two problems with the previous sort of approach, which is that it's computationally inefficient. That's the first drawback. And it assumes that whenever you see two neurons coactive at the same time, that they just must be sharing a receptive field. There's no sort of statistical test done. So we improve the computational efficiency to a quadratic instead of exponential. Sorry, a factorial, and which you might say that it's still not linear, but actually it's a big change to go from factorial to quadratic. And then the other thing that we do is we introduce hypothesis testing using something called information geometry, which I'm probably going to entirely skip what that is, but it's just a way to do statistics, a way to not assume that all the activity is deterministic. And just as a small example, with 25 code words and 46 neurons, for the original neuron, this takes, for the original algorithm, this takes about 3 minutes on a fairly powerful PC, whereas our algorithm takes less than a second. And this is mostly due to the fact that there are far fewer operations to perform.
Sorry, what is the algorithm computing here?
The algorithm is computing basically the same information as the canonical form. It basically calculates generators of the canonical form of the neural ideal of the code, and through that is able to do the tests for these different applications about like convexity of the code, And if it's convex, the lower bound and the number of dimensions that these receptor fields must be embedded within, and things about the geometry or topology of the code that's implied thereafter. And then on top of that, we do the information geometry. So actually, this thing about the number of operations, this is actually just about computing that stuff, and then we do the statistics on that as well, which is extra work, but at least we have a way of doing it. So maybe we could load the last few slides and then.
Cool. Yeah, let's head back to the beginning. Or we can just put them over these 4 if you want, or shall we return to the
sure.
beginning?
Yep, that's fine.
All right, here goes. Okay, the first one is up.
Actually, probably we just skip these ones.
Okay, start with that one. Start with that one. Okay, I'll put that back on the left-hand board then, just to make sure we fit.
Yeah. So this one on the left with the title slide, simulated 2D navigation. This is kind of going all the way back to the beginning, except now instead of a toy example, we have a simulation of this rat in a box with 40 place cells. So here you can see on the left a 2D Gaussian is shown for like 1 place cell, and then there's a red dot with the simulated rodent location, and these purple dots are its previous trajectory. And a shout out to Tom George, who led the production of this nice software package called Rat in a Box, which you can do these simulations in. And in this example, we have 40 place cells, so there are 40 different vertices in this simplicial complex in the middle. And you can see quite nicely that there's a hole in the middle, which corresponds to a hole in the middle here. And then we did the simulation for different amounts of times, like the rat running. Yeah, and then we just simulated for different amounts of times, and using our methods, we're able to compute how many holes do you think this thing has, and it turns out after about 20 minutes of running around randomly, the rat can figure out there's a hole in the environment. Probably it does that, ecologically speaking, much more quickly, but we're giving a very limited amount of information about this. And there are some other sorts of statistics on the right side, which show statistics about this code and the algebraic representation about that. But yeah, then we also do an example with MNIST, a multi-layer perceptron for MNIST. I wanted to show these 2 slides. Okay, it doesn't want to do that. So yeah, we train a multi-layer perceptron on MNIST in a regular way, as you might sort of guess, and then we do the same sort of analysis as shown previously on the 2D environment, but we binarize the activity of the neurons in the second layer, so the penultimate layer before like just giving the 10 classes of the digits, and we look at different epochs in training. So there are 10 epochs that we do, and we then just count things about, and we look at the distribution of like the coactive neurons, so those are those histograms. We also look at the number of monomials or mixed monomials. You notice there are pretty dramatic changes here. The general trend seems to be that you start looking at very high sort of mixes of high dimensional stuff, and then you slowly sort of specialize and things sort of separate and become, the sort of redundancy of the information changes, or it gets lower. I've sort of thought about this a bit like an Easter egg hunt. So the kids, the receptive fields in this, in this hidden layer, are all going for the most obvious things first to differentiate the different classes. Or after one epoch, they have lots of mixtures of sort of overlaps of receptive fields, and there are things which there are receptive fields which just never talk to each other because they've sort of specialized. But then over time, you get a slow mixing of these things. So you go from these disjoint pairs down to zero, and that corresponds with the monomials going down. But then these mixed monomials also go down. So it means that you get less redundant information as well. So then once you've looked for the most obvious places for the Easter eggs, you start looking for the less obvious places in order to capture all the Easter eggs.
I don't really understand what these stats are saying. So I'm supposed to be thinking of a kind of simplicial complex where like the edges sort of say, I mean, if in the second layer, well, so I don't even really understand what the analog of vertices are, I guess. So
vertices here would be, so this is a simplicial complex made from the coactivity of neurons in the second hidden layer, binarized. And a vertex here represents activity of that neuron. And if you have like an edge between those two neurons in this second layer, which obviously doesn't exist in the model, but it exists in our simplicial complex, then that means that those two neurons were coactive at some point.
Right. So.
So we're still looking at some sort of like coactivity of so which of these hidden neurons were coactive together given particular samples. So we put one sample through one at a time, and we look at the coactivity at each time, and then at the end of that we have an example of like, I don't know, not 10,000, but maybe like 1,000 or 5,000 examples of these coactivities, and then we analyze those coactivities. And everything on this slide has already gone through the statistical test, so any statistic here is significant according to the information geometry tests that we did.
Interesting. Yeah, I guess I don't really understand what any of these numbers, or what would be surprising or not, but I suppose my takeaway from this slide is your code is fast enough to run on MNIST.
Yeah. I mean, our code is not necessarily limited by the input, but rather like the number of neurons or the number of code words. So the number of example images or the number of like hidden neurons that you're trying to analyze. So theoretically, it can be as sophisticated as you like, the model or the inputs. What matters most is the number of neurons and the number of things you want to analyze. And in a very vague way, because probably haven't explained it well. What we're doing is we're looking at the coactivation of neurons. We're seeing do they regularly encode for similar things or not, and if they don't encode for similar things a lot, then that's going to be like a monomial relationship. But if they have redundant information, that's going to be a mixed monomial relationship. And then also, if we want to look at the space that these things could minimally, like a lower bound in terms of the amount of space, if we were to embed all these receptive fields, and if they were convex, what's the space they could be embedded in? Well, the more complicated the task, the higher dimension you would expect that lower bound to be, perhaps. And so the fact that it goes down during training implies that it's sort of learning those. The fact that it changes at all during training, I suppose, sort of implies that the nonlinear sort of essentially
Can't really hear you, Tom.
mapped into those. Oh,
I think
sorry.
it's better now.
You're cutting out.
Sorry.
I think
Okay.
it's better.
Yeah. Anyway, maybe I've talked long enough, but yeah.
Cool. So
Yeah.
well, please finish. So you've got your final slide.
Yeah. I suppose, just the take-home message is our algorithm performs pretty well, and we can use that to study the algebraic or topological or geometric structures in artificial and natural biological neural networks.
Yeah, thanks a lot.
That's it.
Cool.
Yeah, I find these techniques and these neural codes very interesting. I guess it's never quite clear to me if there's. I mean, I suppose that's your results are making the claim that there is information already at the level of these structures. I mean, you could say that, well, yeah, of course, the coactivity of neurons is how information is stored, but it may be stored at such a high level where it's so many neurons and their relative firing rates that it's just the wrong level of analysis to attempt to do the kind of thing you're discussing here. But I suppose that's an empirical question, and maybe you would say that it is. I mean, how many neurons do you actually have to be reading from to, in a real rat, produce something like what you were talking about in the earlier slide? I mean, are we talking about 10, 100, 1,000, a million? Like, what is the relevant level of analysis?
Yeah, I mean, presumably you could subsample or sample for some smaller collection and not have to sample from the whole thing. And I don't know actually what you would have to sample. I suppose it's an empirical question, or there's probably some theory to apply as well, but I haven't done that. And I sort of I would say that probably there's multiple things. Like this might just be one level, and then there's also other levels of computation happening as well.
Mm-hmm. Yeah, I guess further questions we can save for Tom next week. And yeah, thanks a lot, Tom, for this series. I think this was an excellent intro to the perhaps to people who haven't looked at this kind of thing before, a sort of surprising range of geometry and algebra and topology that are being used by people to try and understand what's going on in these systems. So I think it's fascinating. And so thanks a lot.
Yeah, thank you, and see you next week.